Uh, Thank you for the introduction and for the opportunity to share our results with the IL-15 transplantation. Briefly, I would like to introduce the interleukin-15 cytokine. This cytokine plays a pivotal role in the innate and adaptive immunity. It acts on the wide range of cell types. Its main function is to promote long-term survival of CD8 positive memory T cells, which in other words means that maintaining the immunological memory. So in a case of reinfection, it can provide a faster response. Another function is that it's also crucial for the natural killer cell development. And the main sites of synthesis are dendritic cells and monocytes. Regarding its receptor, the IL-15 receptor is a heterotrimeric receptor consisting of an alpha subunit, which is responsible for the high affinity ligand binding. It's expressed in antigen presenting cells mainly, and it's also worth noting that the IL-15 cytokine binds to the alpha subunit intercellularly and shuttles to the cell surface in this bound form, which provides a more directed and controlled delivery. The other two subunits of this receptor are the beta and gamma C subunits, which are responsible for the signaling. These beta and gamma C subunits are also shared with the IL-2 receptor. The main three pathways are the JAK-STAT, PI3K mTOR, and RAS-MAP kinase pathway, which can be seen here. I wouldn't go into details, but would just highlight this step 5, because we measured the signaling via the sub-5 phosphorylation. And about that more directed and controlled delivery that I spoke about earlier, here is a schematic about the IL-15 transplantation, in which a presenting cell that has expresses the alpha and the IL-15 is presenting the cytokine to the responding cell that is expressing the other two beta and gamma C subunits. After or before transplantation, there is a proteolytic cleavage of the extracellular domain of the alpha subunit, and this alpha subunit, IL-15 beta and gamma C subunit, is internalized into the responding cell. But recently, another method was described, titled transendocytosis, where there was no proteolytic cleavage, but instead the whole alpha subunit with part of the presenting cell's membrane is internalized into the responding cell. A few words about antigen presentation. Antigen presentation is where the MHC2 on antigen presenting cells presents an antigen to the T cell receptor on the target cell. And regarding the signaling, again, I wouldn't go into much detail, just highlight this CD3 zeta subunit, which is again where we measured the signaling activity. So our aims were, can be put into one question: is the antigen presentation required for the IL-15 transplantation to occur? But more in detail, the direct investigation of the IL-15 receptor assembly during transplantation, are the antigen presentation and the IL-15 transplantation independent or coupled processes? Does the antigen presentation affect IL-15 transplantation, and vice versa? Does the IL-15 transplantation affect in any way the antigen presentation? For this, we use the following model system. We have a virally transducted Raji B cell, which expresses the IL-15 alpha subunit. If we treat this B cell with Staphylococcus enterotoxin A superantigen, which binds to the MHC2 and induces the antigen recognition and the immunological signs formation, or IL-15, which induces the IL-15 transpresentation, and mix them with also virally transducted JERCAT T cells, which are expressing the beta subunit and the gamma C subunit, which is expressed endogenously. Then on one hand, the IL-15 transpresentation can occur and the antigen presentation. Then we labeled the relevant receptors and receptor subunits and determined the assembly or the lack of assembly of these subunits and complexes. We also used positive and negative control for first resonance energy transfer, which I will talk about in the next slide. The point is here that we transiently transfected B cells with IL-15 alpha subunit, where an M-cherry fluorescent tag was either on the N terminal or the C terminal part, and labeled the extracellular part with antibody to use as a positive and negative control. And about the method that we use is the first resonance energy transfer. This is a non-radiative transfer of energy, which happens through dipole-dipole interaction between two fluorophores with adequate spectroscopic parameters, and being within each other's 2 to 10 nanometer proximity. And the molecular proximities can be approximated because the transfer efficiency is inversely related to the sixth power of the donor-acceptor distance. We use the intensity-based FRET measurements, but we also used FLIM-FRET, which stands for fluorescence lifetime imaging microscopy. Unlike in the intensity-based FRET measurement, here we only have to measure one variable, which is the donor fluorescent lifetime. And with this simple equation, we can determine the FRET efficiency. For this, we need a donor-only sample lifetime where the acceptor is not present, and a sample where there is a probable acceptor in the presence of the donor. And as can be seen on this decay curves, the donor, which is alone, so it doesn't have any acceptor to give energy to, has a higher lifetime, whereas the samples where there is acceptor, there is energy transfer, therefore there is a lower fluorescent lifetime. In order to determine which receptor or receptor subunit to use as a donor and acceptor, we measure the expression levels and only use we use the lower expression, the one with the lower expression level as a donor. For the first measurement with intensity-based FRET, we measured the assembly of the beta subunit with the alpha. Here we found that the beta and alpha subunit only assemble if we pre-treated the B cells with IL-15, and in the presence of only the superantigen, there was no assembly. And it's also worth highlighting that when both IL-15 transplantation and antigen presentation occurred, there was a slight increase in the assembly. Regarding the other intercellular FRET, which is done by the FLIM-FRET, we investigated the association between the CD3 and the MHC2. Here, not surprisingly, this antigen-presenting complex only assembled in the presence of the superantigen. When there was only IL-15 added to the B cell, there was no assembly. Regarding the cis interactions, on the Raji cell, we measure the FRET between the alpha subunit and the MHC2, which showed high energy transfer efficiency along all the samples, even on lone cells, inside the immunological synapse and even outside the immunological synapse. The other cis interaction was measured between the beta subunit and the CD3 on the T cell. Here, there can be no very low transfer efficiencies can be seen. So there is no connection between these two subunits. We also noticed that during the IL-15 transplantation and the antigen presentation, there is receptor and receptor subunit translocation to the immunological synaptic site, which we calculated by this formula, where we measured the intensity values of the given subunit in the immunological synapse. Basically, took the average intensity in this synapse and outside the synaptic area, and this ratio gives a normalized average pixel intensity in the synapse. And the results are that the majority of the beta subunit during IL-15 transplantation translocates to the synaptic area if the IL-15 is presented, of course. The alpha subunit and the MHC2 translocate seeming jointly as a complex in the presence of either ligand, while the CD3 and the beta is translocate completely independently, only in the presence of their only ligand.